Hello, in this video we will discuss about the Maxam Gilbert sequencing of DNA. So the Maxam Gilbert sequencing of DNA required the radioactive labeling at one end and is used for the DNA sequence. So let's begin to understand. So the required radioactive labeling at one end 5' of the DNA fragment to be sequenced. So in this way, the radioactive labeling is very important in this mechanism. So in this procedure, so the chemical treatment first and then generates a break at a small proportion of one or two of the four nucleotide bases in each of four reactions. So we will use the four reaction containing the guanine, adenine plus guanine, cytosine as well as cytosine plus thymine. So let's begin to understand the sampling. So remember the DNA extraction is a long process we will cover in the next video. A while you can understand this is the sampling will take for blood, for example. Extraction of DNA is the different mechanism via a cell lysis as well as centrifugation, washing. That is the different things we will not understand, but just sample DNA will be extract. After the extraction of the sample DNA double stranded, which that is the preparation of the homogeneous single stranded DNA. Single stranded DNA means the deoxyribose nucleic acid is contain a single stranded. So these contain adenine, thymine, guanine, adenine, cytosine, thymine, adenine, guanine, cytosine is the sequence from 5' to 3'. So addition of the 32 radioactive phosphate labeling group in the 5' phosphate group. So in this way, this will lead to cleavage at specific nucleotides. When the cleavage at specific nucleotides, so in this way, let's begin to understand the 5' will be attached again and again with the segment of the DNA because the phosphate group to the 5' site will be attached again and again. So in the test tube, so the phosphate group are present, which that's contain 32 labeling group. So the pathway for strand cleavage is using the Maxam Gilbert sequencing method we will discuss. So for example, we are taking the thymidine. This thymidine is basically the formula is the ribose, and here is the thymine containing and hydroxyl group are removed to collect connector will bind. So this connector here with the carbon number 3 and carbon number 6, while here carbon number 5, and in this way the addition of the hydrazine. After the addition of the hydrazine, it will be changed the chemical formula due to the addition, and cyclohexylamine when add. So the chemical formula you can understand in this way. So in this way, the ribose sugar, deoxyribose sugar, will be disappear due to the changes of the chain. So this is the beta elimination, and strand will be cleavage by this mechanism. For example, we are taking the example of thymine. But in this case here, you can see the labeling is 32 labeling the isotopes of the phosphate group from 5'. So the cleavage at specific nucleotides we will discuss. So the cleavage, for example, the G reaction, A reaction with some G cleavage, cleavage we will discuss in the underline in the next. So wait. So T reaction with some C cleavage. So underlined we will also learn. And the C reaction. What that means? G reaction, C reaction, and the T reaction. That is basically occur in the PCR, the thermocycler, which that is a cycle contain a phase. This phase is the denaturation of the DNA. After the denaturation at the 92 degrees Celsius temperature, annealing process, so the specific primer will bind to annealing is known as annealing at 52 degrees Celsius, and extension will be occur due to the 72 degrees Celsius temperature of the thermocyclers, the PCR, polymerase chain reaction. So after this, the complete the more and more amplification of this DNA segment will produce different segments. So here is the, you can understand the ATTG8CT AGCC segment, and in this with the cleavage in the G side. So you remember, and the from G reaction, so that is why the different segment, and here is also the different segment due to the A reaction and the G cleavage, the some G cleavage here you can see. So in this way here is, for example, the A reaction, so the A cleavage. So in this way the segment will be separate, and each segment will attach with the labeling radioactive group. So this is the different, here you can understand that is, and I am giving the underline for understanding the G cleavage, G site, the governing site. So here you can see. While in this case, the T reaction here is, you can see, the T reaction with some C cleavage, the underlined. So in this, so the C cleavage will be underlined. I will underline it. So here you can see that different segments will be produced due to the cleavage at the T side cleavage and C cleavage, basically same like a thymidine cleavage, thymine side, which that we had discussed. So here is the a different segment will produce, here you can see the C reaction. So here is the C and C, and you can understand as well. So the C side will be cleavage to produce more segment, and different their molecular weight and the different their size. So that is why the different. So that we will put this all data into the gel electrophoresis with a difference. So let's begin to understand the gel electrophoresis, the SDS page. So in this way, the radiography we will use for the production of light to understand the segment where is. So the segment will move from negative charge to the positive because DNA contain negative charge. So here you can see the guanine is separately we are putting, and in this way the adenine and guanine and thymine, cytosine and cytosine. So we can calculate this segment and reading by this from upper to the lower downward. So here you can see. So this is the reading and this is the sequence. So that is known as the Maxam Gilbert sequencing through radiography and show the color, which that we can identify where is the G are present, A, G, etc. So that is the basically used for the DNA sequencing